Hello, biology. I wanted to go through your new unit notes so that you get an idea of what all your notes are about and get me explaining some of the more crucial bits. So this new unit is all about the cell cycle, mitosis, and meiosis. Basically, it's about how cells live and reproduce, make more of it like themselves. Okay? So here we go. So I'm going to start out by talking about something that was part of our history, which is a long time ago, before we had any really good technology. The people who were called scientists back then, they actually believed in things coming out of like other things. So for example, like they believed eels came from slime at the bottom of rivers and oceans, and they believed maggots arose from decaying meat. Okay, these things were accepted because, well, obviously we didn't have really good technology, and our observation skills back then really weren't all that great. People didn't collect data, they didn't have good experimental designs, and so things really didn't work out really well. And so with these experiments that weren't done properly, they kind of gave rise to this idea that the living things arose spontaneously from other things just because. They couldn't explain it any easier than that. Okay. So I have a video about spontaneous generation. We'll watch that here right now.
Prior to Charles Darwin, scientists had some rather strange ideas concerning how life began. They believed that living organisms came into being rapidly and spontaneously over a period of just a few weeks. The scientific community believed in spontaneous generation for 2,000 years. And it is a stark reminder that even a majority of scientists can be wrong. In his classic 17th century description of spontaneous generation, scientist Jan Baptist von Helmont suggested that mice came from dirty underwear.
Wanneer je een stuk gedragen ondergoed if you put a piece of sweaty underwear in an open-mouthed jar together with a piece of wheat, after 21 days the ferment coming out of the underwear changed the wheat into mice. But what is even more amazing is that the mice are not small or aborted mice, but adult mice emerge.
Another evidence offered for spontaneous generation was the rotting meat experiment. 17th century scientists observed that if meat was placed in an open jar, maggots formed on the meat weeks later. They conjectured that life, in the form of maggots, arose spontaneously from rotting meat. But in 1668, Francesco Redi, an Italian physician and scientist, overturned this idea. He suggested this proof of spontaneous generation was nothing more than contamination of the meat by flies. When flies landed on the rotting meat, they laid their eggs. Over time, these eggs grew into maggots. Later, the maggots changed into flies. When scientist Redi prevented flies from landing on the meat with a piece of cheesecloth, maggots never formed.
Okie doke. So back in the day, we talked about, we didn't have really good ways to do experiments. And this type of red meat experiment where they had meat be decaying, they did that multiple times, but nobody really did anything other than just leave the flask open and saw that maggots appeared. And so this all changed when a guy named like it talked about, Francesco Redi did the red meat experiment in 1668, where he actually started to take some of the scientific method stuff we've learned before by using different variables. He was able to make better observations than other people by having a flask that was open, a flask that was sealed, and a flask that was covered with gauze. Okay? And he noticed that the ones where the flies weren't able to get to the meat, that meat did not form maggots. So this proved spontaneous generation wrong. Another one that came later to better understand that, like, organisms that are tiny don't just come out of nowhere either. So this one is done by Louis Pasteur in 1864. This was the broth experiment. So many of you have probably heard of the term of pasteurization. Louis Pasteur in this experiment is where pasteurization comes from. So what he did was he took some broth and he basically had 2 flasks. He boiled both flasks, and one of them had this weird S-shaped neck on it, and he basically just he boiled the broth so that it would kill anything in it, and then he left the flask as was with the little S-bend curve thing on it. And he noticed that the broth remained organism free. Okay? Nothing grew in it. It just remained as was. And this was because the flask neck here, when bacteria tries to go in, it gets like caught around here and it can't go up and into the broth. In his second test, he did the same thing. He boiled his broth in this weird, like, S-bend flask, but instead of leaving it on there, he removed it. And a little later, he noticed that microorganisms began to grow in that broth. This showed that when you have enough access to something, you get contamination. If you don't, it will remain organism free. And this is why pasteurization works, is because we put a liquid in overheat enough to kill what's ever in it, and then we seal it off so that it is clean to drink, like milk. We do this with milk, and we do this with other liquids as well. Okay? This flask, I believe, still exists somewhere in Europe in a museum, still organism free to this day. Over 100 years later, it's still organism free. Okay? All right. From here on out, here's some learning goals that we got. We're going to be talking about the blue ones in this first half of the unit. In the second half of the unit, we'll talk about the black bullet points. Okay, so these ones we're just going to talk about cell cycle, the steps of it, as well as knowing the difference between mitosis and binary fission. Okay, meiosis will come later. Here's important vocab that we're going to cover in the first part. The ones that are blue and underlined, the ones that are black and not underlined will come later. Okay, so how do we as people go from these tiny little embryonic fetuses in the womb to full-grown adults and, going to school, going to work, doing our daily life things? And the whole answer to that is cell division. Our bodies, as you sit in your chair watching this video, your body can produce up to 25 million or more new cells per second. Okay. Your body's always changing. It's not staying the same. It's constantly changing all the time. And it's all thanks to cell division. But what is cell division? By definition, cell division is simply the making of more cells from already existing cells. Remember, this part, the bottom part, is part of the cell theory. We already know that we need pre-existing, like cells come from pre-existing cells, okay? All right. We also know that we have two types of cells. We covered that before in the third chapter. Talked about we got prokaryotes, which are bacteria, and then we got eukaryotes, which are like animal and plant cells. Both of them go through cell division. It's just that they are different processes. Okay, I'm going to go over the prokaryotic version of cell replication first. Here are the differences for you between prokaryotes and eukaryotes. This is just an overview. So this is not something like if you need a review of the difference between prokaryotes and eukaryotes, it's there for you to read it. Okay? But anyway, the way that bacteria reproduce is by binary fission. All right? It's a simple 4-step process. It's basically your cell gets longer and the DNA inside here replicates. As it gets more and more stretched apart, it starts to pinch off. It completely pinches off, and then the two cells separate once they are like completely contained from each other. Okay. You can even like whittle this down to a 3-step process. DNA replicates, cell elongates, pinches off, and then separates. Okay. So I have a couple animations for us to watch about binary fission. First one shows you an animation of what happens. The other one's just a fun animation of it doing its thing in it. Okay.
Most bacteria reproduce by a process called binary fission. During binary fission, the parental cell divides to form two daughter cells. In the first step of binary fission, the bacterium replicates or copies its chromosome and attaches each identical copy to a separate location on the cell membrane. Unlike eukaryotic cells, bacteria do not need to dissolve a nuclear membrane or assemble a mitotic spindle, making binary fission very quick and efficient. Second, the cell elongates. Since the chromosomes are attached to different locations on the cell membrane, they separate from one another. Third, the bacterium pinches off at the center and forms a septum, a separating wall between the two compartments of the cell. Finally, the cell splits into two new daughter cells. These daughter cells are fully mature bacteria that can grow and, if conditions are right, continue the binary fission process.
Okay. So that was the first one. And here's the second one. So in this animation, you can see that it's a pretty simple process. And it goes pretty fast, right? It's just taking this one cell it starts with and almost it looks like it just pulls apart pretty much and makes two new ones. And if you remember from our previous chapters, we know that bacteria replicates really fast, and this is why, because binary fission is really fast. Okay? And I got a couple follow-up questions here. So, what's the most important step in this process? And the answer to that is replicating and the separation of your DNA. DNA is your thing that tells your cell what to do. Without it, it doesn't replicate, it doesn't eat, it doesn't do anything. It needs this to survive. And so replicating it so that your new cell has a copy of it is super important. Okay? And just letting you know, this replication of your DNA is making an exact copy of that DNA, because the ultimate result of binary fission is to create two copies of the exact same cell, which are called daughter cells. Okay? This means that this binary fission process is almost like you taking a photocopy, or like going up to a copy machine and copying the same picture over and over and over again, getting the same picture no matter what. Because that's all that bacteria wants to do, is replicate and make the same like cell of itself. Doesn't want anything different. It wants the same. Okay? But that's binary fission in a nutshell. Now let's move on to talk about eukaryotes, or our cells. So in our cells, we go through a process called mitosis to get new cells. Okay? It's used only in multicellular organisms, okay, or like animal and plant cells. We only do it to replace dead or injured cells. Okay? If you don't have any of those, it's not going to go through mitosis. But if it does, mitosis is happening uncontrollably, guess what? You got cancer. Sorry, you got cancer if that happens. Okay. Here's a quick review of our organelles. It asks which organelle is the most important. Another way to ask this question is, where in the world does your DNA hang out? And hopefully you answered here, in the nucleus. The nucleus is where your DNA hangs out, and this is where it lives all the time. It never leaves this place. Okay, unless it's for cell division. It never leaves here. Okay. Here is, excuse me, a close-up of like what is in the nucleus. So in the nucleus, there's something here called a chromosome here. And it looks like this little X structure, but actually what it is this is your DNA. And it's wound up into these X shapes. Excuse me. As you can see here. DNA we all kind of recognize as this double helix structure, you know, when we're first taught. However, where this is true, when your cell divides, what it needs to do is it needs to compact this DNA, and it noodles it up into these X-shaped chromosomes. Okay? And this chromosome is important. It has different names it has different things on it to represent different important parts. Okay? The whole X is called a chromosome. Each half of it, so one half, is called a chromatid. The thing here that holds them together is called a centromere. And then the outside, like the very end of each of the X tips, are called telomeres. Okay? And I went through that because they are important vocab that you have to remember. Okay? And we're talking about the nucleus and why it's important. because it holds your DNA. DNA, I hope you realize, is actually called deoxyribonucleic acid. I don't expect you to know how to spell this. I do expect you to recognize that this is what DNA's actual name is. And it's not hard to see where your DNA acronym comes from. The D is here. Sorry. Computer's being a little slow. So this would be for the D. The nucleic part of deoxyribonucleic is the N, and this is the A. Okay, that's where those come from. That's where how DNA gets its, how we refer to it. Okay. In order to divide, our eukaryotes need to replicate our DNA, and when it replicates it, it forms them into those chromosomes, these X-shaped things. And I just talked about, so chromosomes are what the X shapes are. They're tightly wound and they're ready to go. Chromatin is when your DNA is in the most relaxed phase, its unwound state. So if we go back here, like when DNA is like this and it's all spread out and noodly, that's what chromatin is. Okay. And then chromatins I talked about is there are two exact copies of the chromosome attached together. Like I said, each side of the X is the chromatid. So the genes on this half are the same as the genes on this half. Okay? And that's important later, and we'll talk about that. And then finally, the thing that connects the two chromatids together is called the centromere. Okay? Here's a little cartoon talking about chromatin and how it looks when we look at it compared to chromosomes. So this one chromosome's like, dude, mitosis starts in 5 minutes. I can't believe you're not condensed yet. Meaning that this is our DNA when it's relaxed and not ready to like go through the division process. They turn out like this when they are going to go through mitosis. Okay? All right. I'm going to go through the life cycle real quick here before I'm done. So we're almost finished with this set of notes. So for our eukaryotes, our life cycle for each of our cells consists of 3 phases. Interphase is the first one. Mitosis is the second one. This is where we have the actual division of our nucleus. And then we have cytokinesis, where we have the division of cells from each other when they're being made. Okay, and each cell goes through these steps. Some of them go through it only once. Others will go through it many, many times. An example of ones that do it a lot are things like your skin cells, because you often get cut or, get damaged on the outside of your skin, like you get bruises or like cuts and whatnot. And if you leave it alone, you get better, right? So that means that those cells go through mitosis to get rid of the damaged ones and heal up. Cells that don't go through this a lot, maybe only once in their lifetime or once or twice, are going to be things like your heart cells, because they're with you for your life as long as your heart is around. If your heart, if it was doing mitosis all the time, that means that it would have to like get rid of cells that are important for pumping your blood. So those heart muscles don't go through a lot of this 3 phases. Same with your brain cells or your nerve cells. They don't go through this because they need to be ready at all times to send important signals in your brain. And so when you're a baby and you grow, the brain cells that develop then are probably going to be the brain cells that you have for the rest of your life. You might get a few, like when you're growing as a baby before you're born. Yes, you have the, cell cycle happening for those birth cells, but once they're finished, they usually don't do it again. Okay? All right. But these are the 3 phases that we go through in the cell cycle. And the very first phase, like we talked about, is interphase, which is the growing phase. Okay, this is where 90%, so cells will spend 90% of their time in this phase. Okay, and the reason for that is because, like we talked about, your cells aren't going to divide if they don't have anything to do, if they don't have to repair anything or replace something, it's not going to change. So they spend most of their lives in interphase. Okay. During this phase, they usually increase the amount of proteins, they create more cytoplasm, they make more organelles, and they just, in general, grow in size. Okay. During this time frame in interphase, you can have DNA replication, but it would still be in its chromatin or the noodly spaghetti form. Okay. Here's an overall view of the cell cycle, including the last 2 phases, mitosis and cytokinesis. As you see, interphase takes up 90% of that cycle, and it's broken up into little chunks. Your first chunk is called the G1 phase. It's also called the first growth phase. This is where your cell, a major portion of the cell's life is. It grows and it carries out normal functions. Then it has an S phase. The S stands for synthesis. This is where your DNA is replicated. In other words, it makes another copy of the DNA in this phase. You also have new organelles that are made here. Okay. Then we have a G2 phase or section of interphase, which stands for second growth. And this is important because this is where we prepare for division. This is where we're getting like the pieces or staging the cell to get ready to divide. We get microtubules assembled, we get spindles made, we get things all ready so that when we're starting that mitosis phase, we're ready and we're set. Okay? In our whole cell cycle, we also have checkpoints that our cell actually has to check off if it wants to go from one part to another. So for example, we have a checkpoint here in interphase called the G0 checkpoint, and this determines if the cell goes into a phase called G0, which is basically where the cell just doesn't do anything really anymore, or it goes on to DNA synthesis. I believe we have another checkpoint over here with mitosis. It does some checking of your DNA and making sure everything is ready before it starts. If not, it stays in G2 until we're ready. And then we have one more here between the mitosis and cytokinesis phase to talk about separation of cells from each other. Okay? So basically, what you should get out of these 2 slides is that interphase has different chunks, like different things that happen in it. And in order to go from, certain points or like continue on in the cell cycle at certain points, you have checkpoints which act like stop signs, where if the conditions are not met, you do not go past that point for your cell. Okay? All right. That's where I'm going to stop, because next time we'll talk about each step of mitosis, what happens in each, and what each look like. Okay, so we've learned about both binary fission and the start of the eukaryotic cell cycle by going over interphase and the basics of what happens in each of the areas, interphase and mitosis and cytokinesis. Okay, I'll see you next time for going over mitosis. All right, I'll talk to you later, everybody.